Hi everyone, I'm the Plant Propagator and welcome to my channel. If you like my videos and you want to keep on seeing them, it would help me out if you can click on like, share, and subscribe to my channel if you're not already a subscriber. Today I am back in my old university laboratory and I'm excited to share with you that I will continue to have access to my laboratory a little bit longer. I'm still going to try to clear most of the things out of here that I've used over the past 35 years, but I have to save some of it for the next occupant of this facility. So I'm only going to clear out what I'm allowed to, which means it'll be all of my living orchid cultures and plants and things along those lines, and then whatever I feel that the next person won't want. Today what I want to share with you is some of the techniques that I use in the laboratory, and I want to talk with you about measuring the sizes and volumes and masses or weights of different things. And I want to share this with you. This is something that I tend to ask the PhD students when they're taking their general exam, if I'm on their committee for some reason. I like people to have an understanding of sizes and masses and things along those lines in the grand scheme of things. And it's interesting that for the most part people know maybe some of the larger things, but they don't know what some of the smaller sizes are and how they compare one thing to the other. Before we get into that though, I want to talk with you a little bit about the system that we use for measuring masses and volumes and distances and things like that. And in the laboratory we use the metric system, and this is as opposed, at least in the United States, to the English or the imperial system. And the English system is a little bit awkward to work with if you're trying to calculate things in the laboratory. It just can't be done. It's not what it's for. It's still used, but it's really difficult to work with if you're trying to figure out half or a quarter of some volume or some distance or something like that. It can be very challenging. So we measure things in miles and yards and feet and inches, and then volumes are in cups and quarts and gallons and things like that. And it's just, you can't do this in the laboratory. It just takes too long to convert to this. So I want to kind of share with you my thoughts on this, and I really dislike the gallon, the English system or the imperial system. It just and again, you can't use it in the laboratory. And to give you some example of measurements, so this is a gallon, and this is the measurement in the English or the imperial system. And then what you have, as far as you break down what goes into a gallon, and there are 4 quarts in a gallon, there are 2 pints in a quart, there are 4 cups in a quart, or 2 cups in a pint, and then you break it down further with smaller volumes. There are 10 tablespoons in a cup, and guess how many teaspoons in a tablespoon? It is a round amount, but still, these are the volumes and the measurements that I have to use when I'm measuring most of the things in the United States for my sprayer, for my fertilizer or pesticide application. So, how many teaspoons in a tablespoon? 3. And these things are just they're just odd measurements. The only one that's not odd is a quart in a gallon, because the quart means quartile, or 1/4 of, so there's 4 quarts in a gallon. But aside from that, it's very difficult to calculate back and forth. It's just an awful system for working with, and it's the Brits' fault that we are using this. That's where we come from. That's the language that we speak, and it's just an awful system. But I was driving in England a few years ago, and it was a little bit nice to see the speed limit. Occasionally in miles per hour you would see that, and again, it was nice to have that because I'm so used to it here in the United States, but it's just you don't use that system in a laboratory. It's impossible to use. So what I want to do is go over some of really the ease of the metric system, and then I want to talk about some of the sizes of some of the things that we work with, and to show you how easy it is to move within the metric system and how difficult it is to move within the English system. All right, so the metric system is largely, or in my mind, it's largely based on water. So what's right in here is water. And so the interesting thing is, and it's like, well, what is this? Why is it based on water? Well, so when you measure out of here, when you measure a milliliter of water, that weighs exactly 1 gram. That's the basis for this system, all right? And a milliliter is described as a cubic centimeter. And don't turn me off. This is not that hard. We'll get back to that. So a cubic centimeter is a volume, a centimeter is a distance, and a cubic centimeter, a centimeter times a centimeter times a centimeter, is cc is a milliliter, and 1 milliliter weighs exactly 1 gram. And that's how the system is defined, and that makes it so easy. So if I were to measure out 10 milliliters, 10 milliliters of water, I'm measuring this out, and I can tell you exactly that 10 milliliters of water is going to weigh 10 grams. And I'm not a magician, I'm not a genius, that's just how the system is. If I measure out, here we go, this is getting a little harder. Not really, it's the same. All right, so this is, and I just measured it out, 100 milliliters of water, and guess how much 100 milliliters of water weighs? It weighs exactly 100 grams. All right, this is 500 mils, but it's a liter. So if I would measure out a liter of water, that's 1,000 milliliters. And again, this transition, there's milliliters, there's liters, and that would weigh 1,000 milliliters weighs 1,000 grams, which is a kilogram, a kilo. All right, so this is the great thing about the metric system, is that it's really easy to go back and forth. In the laboratory, it makes it really simple to do that, to go back and forth between one unit and the other. At the low end of things, let's talk about distances for a second. So, a meter, we know a meter's about 3 feet, but a meter's about this long, and then 1 millimeter is a thousandth of a meter. All right, so a millimeter is a lot smaller. 1,000th of a millimeter, again, a really small distance that's hard to see, that you can't see, is 1 micron. That, I guess, is the only flaw that I can see in the metric system, is you've got a Greek letter mu that is the precursor, or that is the abbreviation for micrometer, all right, or micron in that size. So sometimes it's hard to find the Greek letter micron. Now when you go to 1,000th of that, that's a nano. So it's a nanometer, a nanometer, and that's a nm. The thing is, the only thing that sometimes gets confusing in the laboratory is you've got the meter distance is m. So if you have something that is a micromole or micrometer distance, another measurement in the laboratory that is U capital M is micromolar. So the capital M is molar and the small m is meter. And I'm sorry if I'm confusing you, but I just had to throw that in as one of my few concerns with the metric system, is that sometimes whether you use a small m or a large m, it's important. Anyway, let's get back to measuring things. So it's really simple to measure in milliliters and in liters, and when you go below that, when you go 1,000th of a milliliter is a microliter, and we've got these nice tools for measuring the really small volumes. So here we go. If this actually measures one mil, all right, so this is a mil, and I know that I just put on the floor, I just put 1 milliliter, which is a gram of water. And then this actually measures even smaller volumes. So this is actually calculated, or this is it's set up for 100 microliters. So this is 100 microliters on the floor, and 100 microliters I know weighs 100 micrograms of water. And it's nice and easy to split things. And you can even test the accuracy of this equipment, of this pipetter. If I would take 100 microliters and weigh it out on that analytical balance right there, I can even test the accuracy and adjust it, because I know that 100 microliters weighs 100 micrograms. I have other instruments that I can use to measure 1 microliter, which is, it's less than a drop. It's a tiny, tiny amount. And I know that that's going to weigh 1 microgram, which that balances can accurately weigh. So this, again, is the beauty of the metric system, is that it lets you go back, it lets you transition, and it lets you measure, you know, a quarter of or a half of, because you just take these nice round numbers as you're adjusting, and you can divide them evenly, and it's really easy to divide and calculate. So if I only need a half a liter, I can divide all the components in half. So instead of 10 mils, I'll use 5 mils. Instead of 5 mils, I'll use 2.5 mils. You know, and if I'm looking at the English system, if I have to use a pint, then half a pint, I have to calculate how many cups in a pint and then figure that out, or half of a cup. You measure half a cup, but a third of a cup. It just makes it really difficult. So that's the metric system, and how great it is to use in the laboratory. And that's why really in all laboratories the metric system is used. What I want to talk about is some distances, or some sizes, and I want to talk about small things, because I want to relate this back to plants and plant cells, plant organelles, other organisms, and then finally orchid seeds. As far as sizes go, these are things that I like to put out in front of people. And again, I said I'd like to ask the PhD students that are ready to graduate if they can tell me the size, for example, of a plant cell. And it's interesting because they struggle with that, because these are things that are important to understand, and if you're working in the metric system, it's a little easier to grasp. All right, so let's talk about a plant cell and how big they are. And plant cells can range in sizes, and depending on the kind of cells that they are. Some plant cells can be huge, but in a typical plant, the cells, like for example in the leaves of a plant, the cells are going to be anywhere from 50 to 100 microns in size. And the cells can, a lot of times plant cells are oblong, and they're not perfectly, you know, cubical, but they tend to look more like, you know, they're just, well, they're elongated structures. They're bound by a cell wall, so in many cases they're square, and that's how they're packed together. So they can be, but when you look at these cells, again, they're anywhere. They can be, you know, 50 microns to 100 microns, and that's typical. And the vacuole inside the cells makes up most of that volume. There are smaller cells, the cells that contribute to all the cells in the plant. They're meristematic cells that are the growing point of the plant, both in the shoot, so in the shoot meristem, and in the root, so in the root meristem. Those cells tend to be smaller, and those cells are rapidly dividing. They tend to be more cytoplasmic, very small vacuole or no vacuole at all. And those cells were going to be smaller, so those cells are only going to be about 20 to 30 microns in size, and those also tend to be more, it's called isodiametric. They tend to be more cubical or spherical. So the cells, again, plant cells overall in the plant can be anywhere from 10 to 20 to 100 microns in size. Okay, let's talk about bacteria relative to a plant cell. How big is a bacterium? And it was funny, I looked up a few things. So I looked up how big is a bacterium. Bacterium is one, bacteria is plural, and when I Googled this, it wanted to auto-correct for how big is a bacteria, which is just terrible because, again, it's a bacterium, and bacteria is plural, but that's another issue. And I want to tell you one more thing. I was going to title this video, How Big Is It? And where I was going to talk about how big the cells and the bacteria are and things like that. And I did a search on YouTube for how big is it, and I think the content of most of those videos was inappropriate for the content of my channel. So this is going to be titled Easy Lab Measurements in order to get away from that. So bacteria, how big are bacteria, or how big is a bacterium? And so bacteria tend to be anywhere, and they're long. Most of them they can be all different shapes and sizes as well, but they tend to be more elongated. So those are about 0.5 to 1 micron, one dimension, and they're about 1 to 10 microns in another dimension. So way smaller than a plant cell. So the bacteria are typically a lot smaller. When you look at the contents of a plant cell, so the two main organelles that are in plant cells are mitochondria and chloroplasts. So when you look at those are believed to be derivatized from bacterial cells. They are about the same size. So they're about 1 by 10 microns, and they're shoved into a plant cell that is anywhere, like I said, from 20 to 100 microns large. So you can get an idea of the scale of things, and knowing these I think is important, and it's what many of the graduate students that are getting their PhDs, some of them know it, some of them don't, but it's interesting. These sizes and the relative sizes are important to know. Okay, so let's talk about things a little bit more. So the plant cells, the big plant cells are 100 microns for the really big plant cells. How big is an orchid seed? And I've mentioned this before, and if you look it up online, you can see that orchid seeds are dust-like, so they're tiny. And because they're so small, you can fit between 10,000, 100,000 to a few million seed in the seed capsules that I've shared with you previously. And I've showed you some before that I think are a million or maybe 2 million seed, and they're small and they're just like, all right, how big are orchid seeds that you can shove that many of them into a seed capsule? Okay, anyone have an idea? Anyone guess? Well, orchid seeds are they're about a millimeter in length. They can be a little smaller, they can be a little bigger depending on how things are. All right, so and when you look at this, it's interesting because you've got a millimeter, and we said that the average plant cell is 50 to 100 microns, but the embryo, I'm sorry if I'm getting too much detail, the embryo that is in the orchid seed, it consists of really small cells, and these are these meristematic cells. So the embryo it's very compact cells. They're meristematic cells that make up the orchid embryo. And so the cells that are in orchid embryos are probably 10 to 20 microns in size, and that way you can fit a few hundred of them in the seed that ends up being 1 millimeter in size itself. All right, so those are some things, again, to have an idea of scale. So you got bacteria at the small end of things, the plant cells, and as you might imagine, when you put a few hundred plant cells together to make an embryo, that's the larger unit. That's going to be about in the case of orchid seeds, it's going to be about a millimeter or a little bit larger than that. So that's all I have for today. I hope that you stayed with me. I hope that was of little interest to you at least. And I hope you learned something today. So that's all I have for today. Again, I hope you enjoyed it, and happy propagating.